So this video is going to go over learning objectives 2 and 3, and then I'll have a final third video going over learning objective 4. All right, so let's talk about the central nervous system synapses. You are familiar with the term synapse from the neuromuscular junction lecture, or you should be by now. And so that synapse is that functional connection between when in the neuromuscular junction, the synapse was between an axon terminal of a motor neuron and the skeletal muscle cell itself. But in the CNS, these synapses are going to be between, here's some terminology, a presynaptic neuron, which is going to be sending information down to its axon terminals, which will then synapse with a postsynaptic neuron. So presynaptic neuron sending information to the synapses of a postsynaptic neuron, right? So a synapse is that structure that permits a neuron to pass electrical or chemical signals from one neuron to the next, okay? All right. And I put this image over here just to show you that a lot of times when you think about a synapse between two neurons, you're going to think about these axon terminals of the presynaptic neuron coming down and synapsing on the cell body as well as the dendrites. And that's because we just learned that the cell body or the soma and the dendrites are the receiving end, right? So that's where they're going to receive impulses from other neurons, those presynaptic neurons. But you can also have synapses down on right near the axon terminals, and this helps regulate. It could potentially inhibit certain signals from being sent. But I just wanted to show you that there are differences. There's axodendritic, there's axosomatic, there's axoaxonic. So there are different synapses, but most of the time, typically what we're thinking about are going to be presynaptic neurons synapsing with dendrites and or cell bodies. Okay. All right, so there are two types of synapses. There are electrical synapses and chemical synapses. I'll just briefly discuss what electrical synapses are first, because they're rarely found in the CNS. You will see electrical synapses between cardiomyocytes as well as smooth muscle cells, which are muscle cells that are capable of conducting action potentials, but there are few examples of electrical synapses within the CNS. All right, so what are electrical synapses? They are formed between, so here we have a presynaptic terminal and a postsynaptic, so presynaptic, postsynaptic cell. And these two cells are joined by channels, right? These are gap junctions that allow for it pretty much allows the two cells to share cytoplasm. And so when one cell is conducting an action potential, that electrical current can very quickly pass from one cell to the other because the ions can freely flow back and forth, right? So electrical synapses are formed by gap junctions that allows these two cells to share cytoplasm, allows ions to freely flow from one cell to the other back and forth. And the fact that the ions can flow back and forth, this means that the transmission is bidirectional. It can go in either direction. And the main function of those electrical synapses is to allow for nearly instantaneous transmission, right? So within 0.1 milliseconds, we have an action potential being transmitted from one cell to the next. And we'll see next in the chemical synapses, which you are familiar with from the neuromuscular junction, that transmission takes 1 or more milliseconds, right? So nearly instantaneous transmission of action potentials because we're essentially sharing cytoplasm. So they're bidirectional, they can go either direction, and it's nearly instantaneous. So you'll have these anywhere where you need really synchronous excitation of neurons. Now, most of the synapses in the CNS, and the one that you're most familiar with because we talked about this in the neuromuscular junction, is a chemical synapse. And with chemical synapses, there's a one-way transmission of the signal, and that's because, so here, let's orient you with this figure, we have our presynaptic axon terminal, and I know that this is presynaptic without even looking at the little label right here, because what do I see? Just like in the neuromuscular junction, I see vesicles containing neurotransmitter, and lots of mitochondria, because we have all the synthesis of neurotransmitter happening in the cytoplasm there. So this is the presynaptic terminal. And then here, I know this is the postsynaptic terminal because I don't see neurotransmitter vesicles, but I see lots of neurotransmitter receptors embedded in the plasma membrane, right? And so some of these, I've already hit on the fact that you see synaptic vesicles with neurotransmitters, lots of mitochondria because of the synthesis. Just like we saw in the neuromuscular junction, the fluid-filled space between the presynaptic and the postsynaptic cells, this is our synaptic cleft. It's where the neurotransmitters, that space that the neurotransmitter is going to diffuse across. And then we have our neurotransmitter receptors embedded in that postsynaptic membrane. Okay. So these types of synapses are far more efficient. You don't get loss of signal, but there is a little bit of a delay because we have to have all these events taking place before we can get the postsynaptic cell being either excited or inhibited, right? Because we have to have vesicles fusing and exocytosis and then binding of receptors, so it's not as instantaneous as what you see in the electrical synapses. Okay, but they're far more efficient. And I just want to highlight the fact again that it's a one-way conduction at chemical synapses, whereas at electrical synapses we had bidirectional transmission. This is one-way transmission, so it really makes sure that signals are sent. There's a directionality to it, and signals are sent exactly to the location where it needs to be, and it carries out very specific discrete functions. I'm going to refresh your memory about what happens, the events at a chemical synapse, but this is repeat from our neuromuscular junction lecture. So what's going to happen is, here's our presynaptic neuron axon terminal with our synaptic vesicles. An action potential is going to travel down the axon of the presynaptic neuron. It's going to arrive at this axon terminal, causing voltage-gated calcium channels to open. And because there's a higher concentration of calcium in the extracellular fluid compared to the intracellular fluid, calcium is going to rush in. There's going to be an influx of calcium following its concentration gradient, causing more depolarization within the axon terminal, but also calcium is going to cause the docking of the synaptic vesicles. When they dock, it's going to undergo exocytosis, dump the neurotransmitter into the synaptic cleft. That neurotransmitter is going to diffuse across the synaptic cleft and bind to those neurotransmitter receptors that are only located in the postsynaptic cell, right? So the fact that neurotransmitter vesicles are only located in the presynaptic terminal and the neurotransmitter receptors are only located in the postsynaptic membrane, that is what makes it highly efficient, as well as one direction. The signal can only go from presynaptic to postsynaptic. Okay. Once that neurotransmitter is bound to the receptors on the postsynaptic cell membrane, depending on what type of receptor it is, will determine the events that take place on the postsynaptic membrane. We'll get into more of that later. All right, so let's cover learning objective number 3 now. And I'm only going to very briefly kind of talk about the different neurotransmitters. There could be a whole lecture or more just on this. There are 2 major groups of neurotransmitter molecules. There's small molecules, which are going to, exactly what it sounds like. You've already learned one of the small molecule neurotransmitter, which is acetylcholine. We talked about that in depth in the neuromuscular junction. And then small molecules are very rapidly acting and have very acute short-term responses, elicit short-term responses, whereas neuropeptides, which the definition of a peptide is 2 or more amino acids linked together through peptide bonds, right? But neuropeptides are going to be larger molecules, and their synthesis takes longer, their arrival at the axon terminal takes longer. So they're slow acting, but they cause more profound long-term responses. So here, this is just, I'm not going to add, I'll bring up a few of the really important small molecule neurotransmitters, but I'm not going to ask you to memorize this table by any means. But this should sound familiar because when we talked about acetylcholine with the neuromuscular junction, all of that, what we talked about in regards to acetylcholine, is the same for other small molecule neurotransmitters, and the fact that these small molecule neurotransmitters are going to be synthesized in the cytosol of the presynaptic terminal, so they're constantly being synthesized there, then packaged into those synaptic vesicles, and they're right there for immediate release upon an action potential arriving and those voltage-gated calcium channels opening. These small molecules very rapidly diffuse across the synapse, and they're very fast-acting and have very acute short-term effects on the postsynaptic membrane. Okay? And then the vesicles and the neurotransmitters are continually recycled very quickly. So now, just if you remember, we talked about acetylcholine. In the neuromuscular junction, acetylcholine is always excitatory, but when we talked about, remember, in smooth muscle, acetylcholine or norepinephrine, they could either be excitatory or inhibitory, depending on the type of receptor that it's binding to. Let's see, glutamate is the big excitatory neurotransmitter in the CNS, whereas GABA is the major inhibitory neurotransmitter in the CNS. So those two, glutamate and GABA, you've probably heard about maybe once or twice. Glycine is usually inhibitory and usually primarily in the spinal cord. So again, I'm not going to ask. There are other important things that I can ask about, but acetylcholine you should for sure know. Okay, neuropeptides, they are synthesized not in the axon terminal, but they're going to be synthesized in the cell body where all the protein machinery is. So right where like the nucleus is for transcription, and the rough endoplasmic reticulum and the Golgi, all of those big areas that are necessary for protein synthesis, that's where neuropeptides have to be synthesized. And they're usually synthesized as larger protein precursor molecules, and then they get chopped down and further modified into the active form of the neuropeptides. All right, so they're synthesized in the cell body, so then they have to be packaged into vesicles, and they have to travel all the way down the axon, eventually getting to the axon terminal. So it's a much longer process for these to be synthesized and transported to the axon terminal. But once released and once they arrive at their site, they have much longer, more prolonged effects to make up for how slowly they're synthesized and how slowly it takes to get there. Then they have much more prolonged effects on the postsynaptic cell. And these guys are not reused and recycled. If you look at the table over here, some of the most common neuropeptides, you've probably heard of several of them in regards of being hormones like thyrotropin and growth hormone, glucagon, insulin, calcitonin, right? So these are all neuropeptides. So sometimes referred to as like hormones, and that's because they have much more prolonged effects. Oops. All right, there are two major types of receptors that can bind neurotransmitters, and one class. It's called ionotropic receptors, and these guys, the reason why they're called ionotropic is because once a neurotransmitter binds to an ionotropic receptor, it's an ion channel. It's an ionotropic ion. Right? It's an ion channel that very quickly changes conformation, opens, and then allows for the passage of ions into or out of the cell. So much quicker. Okay, directly controls the movement of ions in or out of the neuron. So that's ionotropic. Metabotropic receptors are receptors that once the neurotransmitter binds, right, it doesn't directly open this receptor, but it causes metabolism changes within the cell. Okay, so you have the neurotransmitter binding to the metabotropic receptor, which in turn is going to activate a G protein, which will then activate a second messenger cascade system that can exert wide effects on the postsynaptic cell. Okay, and I'll go in a little more details right now. All right, so ionotropic receptors. Again, the neurotransmitter, once bound, has a direct effect on the conformation of the ion channel, opening it or closing it. I guess it depends on what the ion channel is, but it's going to directly control the flow of ions into or out of the cell, right? If we are talking about, this is just some like terminology type stuff. Hopefully everyone's familiar with, if you hear the term cation, it's referring to charged ions, or anions, that is referring to negatively charged ions. And if you know, so this is going to go back to your action potential lectures, I'm sure, one of your first lectures from last week. But if you know that the resting membrane potential of neurons is negative, about negative 65-ish, right? So the resting membrane potential is about negative 65-ish, give or take, right? And then if you know where the most important ions have their highest concentration, so for example, if you know sodium ions you have a higher concentration of sodium ions in the extracellular fluid. You have a higher concentration of calcium ions in the extracellular fluid. You have a greater concentration of negatively charged chloride ions in the extracellular fluid. In the intracellular fluid, you have a higher concentration of potassium ions. So if you know where these major ions are located and where they have higher concentrations, if you're given a question or a situation where a sodium channel opens, you know that sodium is going to follow its concentration gradient, and the fact that it's positive, it's going to want to go, the positive is going to be attracted to the negative resting membrane potential. So there's a concentration gradient as well as an electrical gradient for sodium to rush into the cell, right? If you're given a situation where a chloride ion channel opens, right? Well, you know chloride ions have a higher concentration in the extracellular fluid. That concentration gradient is going to want to bring chloride ions into the cell. If a potassium channel opens, you know that potassium, it has a higher concentration on the inside of the cell, so potassium's going to want to leave. So I went into that just so I find, instead of just memorizing and compartmentalizing information, if I just know big picture stuff, I can apply it to other questions, okay? For example, if there were to be a question about ionotropic receptors being opened and what would happen as a result of that being opened, you could apply those facts, that information to help answer it, if that makes sense. All right. Let's see. And I'll go over this a little bit more in the next video that I make. But if there's an influx of positively charged ions into the postsynaptic cell, right, that's going to cause depolarization and it's going to excite the postsynaptic neuron. If there's an influx of negatively charged ions, like chloride ions, that's going to make the resting membrane potential more negative. It's going to cause hyperpolarization, and therefore it's going to inhibit the postsynaptic neuron. I'll talk more about this later. So what's the major point here? Ionotropic receptors, once bound by a neurotransmitter, that neurotransmitter will directly cause or influence the movement of ions through that receptor, through that channel. Whereas in metabotropic receptors, so here we have our metabotropic receptor, a neurotransmitter binds, and then this does not directly open, right? So we don't get movement of ions right away, but we have the activation of a G protein, which can then go and affect ion channels, and we can get movement of ions. We can also get activation of enzymes that can synthesize very quickly second messengers like cyclic AMP or cyclic GMP. These second messengers can then go and affect different intracellular proteins in lots of different pathways. We can get activation of gene transcription, and therefore we get changes at a transcription and translational level with different proteins being synthesized or turned off. So with the metabotropic receptors, we get an amplified intracellular response and a much more far-reaching response cellularly, right? And if we're getting transcriptional changes, that can have much longer term effects on the cell versus just opening or closing of ion channels, okay? And then at some point we have to have termination of this signal, right? Of the transmission from our presynaptic neuron to the postsynaptic neuron. We have to be able to turn it off at some point. And how that's going to happen is, if we're talking about like small molecule neurotransmitters, we can have reuptake into the presynaptic neuron, and they can be repackaged into vesicles and reused. So we can have reuptake. We can have degradation by enzymes that are located in the synaptic cleft. An example of that we learned in the neuromuscular junction with acetylcholinesterase, which is there to very quickly degrade acetylcholine. And we can also have diffusion of neurotransmitters away from the synapse, and all of that can help terminate the signal because we have to have it stop at some point. Go over this again. So the fact that I've gone over this in neuromuscular junction lecture, I've gone over it once already, and I'm going to do it again. You should know the events of synaptic transmission. First, we have an action potential arriving at that axon terminal, causing depolarization and opening of voltage-gated calcium channels. Calcium has a higher concentration in the extracellular fluid, so we're going to have calcium influx into the synaptic terminal. That's going to cause more depolarization as well as docking and fusion of our synaptic vesicles. We get exocytosis and dumping of the neurotransmitter into the synaptic cleft. Then it will diffuse across the synaptic cleft and bind to receptors on the postsynaptic membrane. These receptors can be ionotropic receptors, which will directly influence the movement of ions into and out of the cell, or these could be metabotropic receptors, which will then elicit a second messenger cascade and have far-reaching effects on the postsynaptic cell. Okay? And as far as whether or not we will get excitation or inhibition on the postsynaptic cell, I'll go into that in our next lecture, or the next video for this lecture. But this blue box right here highlights an important concept, is that the quantity, how much neurotransmitter gets released into the synaptic cleft, is going to be directly related to the amount of calcium ions that enter into the synaptic terminal. So the more calcium we can get influxing into the synaptic terminal, the more exocytosis we get, the more neurotransmitter into the synapse we get. And then the last little video will go over our final objective for this lecture.